Hello everyone, my name is Rich Ottinger and I am the Marketing Programs Manager for Park Systems. Welcome to the 6th installment of Park Systems 2019 Material Science Research and AFM webinar series. Today's presentation is titled Polymers in Medicine, an Introduction. Before we begin, let me give you a quick overview of today's session. The presentation is expected to take approximately 40 to 45 minutes, which should allow us some time for a question and answer period at the end. If you click the raise your hand button at that time, I can unmute your line to ask a question. If you would prefer, you may type in your questions at any time during the webinar, and I will try to pose these at the end. Any of these questions that we do not get to, we will try to address in a follow-up email. It is now my pleasure to introduce to you Dr. Rigoberto Advincula. Dr. Advincula is a professor of Macromolecular Science and Engineering at Case Western Reserve University and the editor-in-chief of MRS Communications. He is a fellow of the American Chemical Society and is the author of more than 250 peer-reviewed publications. Please welcome Dr. Rigoberto Advincula.
Well, thank you, Richard. Again, I'd like to thank our audience. Good morning, good afternoon, good evening, wherever you are. Today, a very interesting topic. We're going to review polymers in medicine. Now, I changed the title to Polymers in Medicine and Pharmacy because a lot of qualifiers can be made on what is called medicine and what is called pharmacy. But for those of you who are here and are interested for pharmaceutical applications, you're at the right place. So Case Western Reserve University, I'm a professor with the Macromolecular Science Engineering Department. Very nice time of the year at our place. Cleveland Museum of Art, Cleveland Orchestra, nice evening. Nice day to visit anytime with the museum, which is for free. As I said, I'm with the Macromolecular Science Engineering Department. I'm a full professor there. Here's a picture of some of my crew members, and we're located at the 5th floor of the department. So a lot of things we do involve interfacial chemistry, patterning, polymerization, different types of nanostructuring. And today I'm going to talk more about our interest in polymers for biomedical engineering, materials, and even drug delivery. So a lot of our previous publications have actually been centered on DNA, polymer excipients, different types of polyethylene glycol or PEG derivatives, antimicrobial agents, and so on. In fact, at the end of the talk, with the remaining time, I'd like to show you some of our work on polymers for biosensing, for those of you who will be interested to stay. But today will be quite different because this is also a tutorial and introductory talk. Okay, so if you end up going to a drugstore or a pharmacy, certainly other than drugs, you don't expect to see polymers anywhere around you, but you may see things in the form of diapers, personal care, packaging for medicine, and even hairspray. So what I want to emphasize is that all over us, everyday things, even a visit in a pharmacy will allow you to learn more about polymers, whether it's diapers consisting of polyethylene and polyacrylic acid for the absorbent, buying a toothbrush with nylon and polyethylene, or shampoo containing different types of viscosifiers as silicone, or hairspray with polyvinyl pyrrolidone. There's lots to learn about polymers being used all over. And in particular, when you take a laxative, you actually have polyethylene oxide, PEO, as part of the composition or mixture. A drug tablet will actually contain pharmaceutical polymer excipients such as cellulose acetate, starch, polylactides, and so on. So these are things that we take for granted, and we sometimes see them visually, and sometimes we just ingest them. Now, what is the state of the art? Well, we can talk about nanomaterials, nanotechnology, and certainly polymer materials, and I consider them a lot as true compositions comparable to other types of nanomaterial. Can be found in things like tissue engineering, tissue regeneration, scaffolds, bioresorbable materials, or even polymers as drug carrier systems. We're going to look at micelles, dendrimers, and liposomes later, or even polymer drug conjugates. As far as molecular diagnostics, are a contraction of two terms called theranostics, therapy and diagnostics. There's a lot of polymers that are used to enhance imaging and targeting, whether it's protein-protein interaction or signal transduction or intercellular trafficking. Certainly with biosensors and biolabels, that this can be in the form of materials that are introduced or different types of biomaterials that are found in transducers. And certainly a big word is drug discovery, or ways by which one can enhance or high throughput drug discovery through identification of biomarkers, specific diseases. And beyond this, there are many applications of nanosystems and polymers, in particular for detection of pathogens, detoxification, separation, purification, and so on. So what I'm trying to drive is that this 1 hour talk or less is not enough really to cover what will comprise of nanotechnology and polymers. And I use them interchangeably here at this point because in many ways, polymers have played a role in what we call nanotechnology today. So what I wanted to show is in this collage, you can see that polymers as a regime falls in the nanometer to 1,000 nanometer or micron range, or micrometer range. In that, one can be talking about vesicles or dendrimers, or in combination of encapsulation or microparticles. We could be using natural polymers such as proteins, DNA, or even synthetic polymers like polylactides or PEO. The construction or the constitution are both a function of hierarchy of structures, in that you can use self-assembly to improve their efficacy, or to use them as scaffolds in a more network or hydrogel structures. We are interchangeably talking about polymers and nanotechnology at this point, and that could be the subject of a lecture, another lecture by itself. Now, what's interesting with polymers is that you can actually synthesize different types of architectures or microstructures, ranging from homopolymers to copolymers. So copolymers means that we can have random, diblock, or graph copolymers, or we can even have hyperbranch and dendrimeric systems. Certainly, these are closer to nanomaterials or nanoreactors that have actually been used for drug delivery or improving cell targeting. Now, polymers as drug carriers has many benefits in terms of controlled release. Now, the term polymer excipient, which we will discuss in a little while, refers to actually the help of polymers in packaging the drugs so that you can improve their efficacy. However, polymers can be used also for encapsulation, in that you can control or trigger the release upon the proper stimuli, or in the form of gels that can contract or collapse, and therefore releasing the drugs that are contained in those packaging or delivery agents. Protein polymer hybrids, peptide aggregation, targeted drug delivery. Again, I'm just going through a number of terms here because this type of hybrid polymer systems are used to enhance their physiological benefits when introduced to the human body. Of course, peptide drugs, polynucleotide drugs are a classification of itself, which can be used for other types of therapeutic or more disease-specific targeting that can be discussed in details. Now, some of you probably came here thinking, well, polymers in medicine definitely should talk about implants, surgical implants, sutures, artificial devices, artificial heart, breast implants, and so on. You are correct. Polymers are abundant in terms of sutures, surgical devices, tissue, engineered tissues that can be used to prolong life. And of course, we're not even talking about prosthesis or orthesis devices. So for example, for their mechanical properties and inertness, even the polyethylene and polypropylenes are widely used. PMMA, of course, has been well known in the past for their contact lens application, whereas these days there's a lot of compositions based on silicones. PLA, PGA are copolymers that are used for sutures simply because they have the thermomechanical strength, but they also degrade over time or very compatible with wound healing. And certainly silicones, polydimethyl siloxanes, various compositions thereof are used pretty much in a lot of breast implant and cosmetic surgery and augmentation as well. So again, polymers in medicine can be quite abundant when seen in the operating room. Now the challenge, of course, is once these polymers are used in surgery, not only are they classified as biocompatible, but they should have the specific thermomechanical properties to function inside the human body, meaning they should have the elasticity, the tensile strength, and of course the surface functionality so as to prevent, let's say, biofouling or even blood compatibility or preventing any type of thrombosis occurring, specifically when used with surgery in blood vessels. Okay, so polymers, or if we say plastics, that include the polyolefins like polypropylene, the Teflon. We can be talking about polyesters and polysiloxanes. Actually, a big polymer out there that is used in a lot of surgical implements are polyurethanes, and certainly they are processed or shaped so that they can be built into devices that are used for surgery. Now, other than that, we can be talking about polymers that are used actually in ion exchange resins that can be used for purifying blood. A lot of things that we take for granted with dialysis has to do actually with polymeric resins that are meant to be very compatible and high throughput when it comes to dialysis. Okay, specific polymers or polyelectrolytes for absorbing different types of biomolecules for separation system and so on. Okay, how about drug delivery? Can we talk more about drug delivery? Many drugs, their potency and therapeutic effects are somewhat limited because of their solubility or lack of targeting. Once ingested, sometimes we just assume that they will approach their target. However, polymers have played a very important role with polymer excipients. And again, we're going to talk about that in a little while. But at the same time, polymers can be used for targeting, and specifically with non-viral gene delivery systems or even bioconjugates. And this is the case where the specific architecture or design of dendrimers, block copolymers, or even surfactants that can be used for liposomal delivery play a very important role in advances in drug delivery. Okay, I know that some of the slides are very wordy, but just be patient and keep in mind that some of these things actually can be meant for you to read and review later as well. So of course, the goal of a polymer synthetic chemist is to come up with different architectures that can be used as an active ingredient or even as a passive ingredient in drug delivery. Now, targeted drug delivery means that we need to have a biomarker or a therapeutic molecule that is more efficient, let's say, in targeting cancer cells. And that means you have to take advantage of some of the non-covalent interactions or cell-to-cell interactions that can be afforded, let's say, by using aptamers and or even oligopeptides that can be part of that specific targeting. So therapeutics sometimes go hand in hand with what we call diagnostics. So the term theranostic means that you combine the targeting and the delivery of the drug as well, or the diagnosis of disease based on specific fluorescent or radio markers that can be detected or imaged with a microscope. Okay, so let's talk about pharmacokinetics. Pharmacokinetics is an important study when dealing with the efficacy of drugs that are used for things as low as analgesics, but can be as high as therapy used for cancer treatment. That is, at the beginning, you have a high dosage which over time is consumed. And depending on the administration, whether it's intravenous, intramuscular, or oral, you can see that the time of administration plays an important role in its release profile or kinetic or efficacy profile. So what we want to do is to hit the right spot as far as pharmacokinetics, in that we want to control the difference between a conventional application and a slow release application. In fact, the reason why you have to take a drug every 4 or 5 hours is because the drug, as the efficacy or amount goes down, it has to be maintained at a specific level. So therefore, every 4 or 6 hours, you need to take the drug. Therefore, anything that allows you to have a controlled release or even a slow release application is always effective in any drug pharmacokinetics. As you can see here, drug serum concentration versus time for slow release, the ideal case is that you're able to maintain the release of a drug over a long period of time. That is, you don't have to take the drug every 4 to 6 hours, but then you can take just one tablet that's good enough for release, let's say for 24 hours. It's because the most effective concentration of a drug, of course, is between a toxic level and a minimum effective level. So when you're trying or being administered a drug, you need to maintain a window by which this drug is released. That is why we talk about dosage and pharmacokinetics. So in a pharmaceutical dosage, what happens is that a polymer, for example, can be added to emulsify the whole composition, and this polymer emulsion or excipient is expected to provide some of this controlled release, quote-unquote, pharmacokinetic cycle. And this can be fulfilled either by synthetic or semi-synthetic or natural polymers even. The good news is natural polymers like cellulose or starch are actually quite abundant and used a lot in extending release or controlling a polymer function. So what is a polymer excipient? Essentially, this is a substance included with the active pharmaceutical ingredient that is used to control the dosage and its form. The polymer can have two main categories. It could be a protective coating polymer, which is enteric and therefore are able to withstand prolonged contact with the gastric fluid. So actually, when you ingest a polymer, it does immediately affect or is affected by the gastric juices, which is acidic, and therefore trying to maintain it is a good function of the coating. On the other hand, a specific controlled release for drug delivery could be a matrix polymer or a polymer conjugate even, that allows the drug in terms of its diffusion kinetics to be more controlled rather than just a packaging or coating event. Now, pharmaceutical excipients actually are used a lot and are studied very well, much with the type of processing conditions for drug preparation, whether it's an emulsified drug or a tablet or enterically coated tablet. Polymer excipients are part of what goes to the whole composition, which would contain flavoring agents, viscosifiers, preservatives, and so on. So polymers play a very important role in any of these compositions. So some of the most common polymer excipients out there can be in the form of celluloses, polyacrylic acid, polyvinyl pyrrolidone, polyvinyl alcohol, and of course, polyethylene oxide. And as you can guess, these polymers have to meet the drug pharmacopoeia and comply with US FDA regulations, and of course, has to have been used and vetted in the right formulated form. They are designed to swell, hydrate, or at the proper osmotic event be released inside the body. Specific types of polymer pharmaceutical excipients can be in the form of gels or gelatin capsules in terms of a compounded starch. This could be starch, derivatized starch, cyclodextrin, maltodextrin, and so on. This could be in the form of cellulose derivatives, which, by the way, is not soluble, unlike your starch. But cellulose can be derivatized, as you can see, in various forms, and some of them are semi-synthetic, in that a lot of these water dispersible matrices are actually used in a lot of polymer drug excipients, and of course very common in many cosmetic applications as well. Now, going towards polymer, synthetic polymers like acrylic polymers, carbomers, pentacrylates, of course polyethylene oxide, polyvinyl alcohol. Some of these have specific uses for improving quality control as well as interaction with the drug, viscosifying effect, and so on. And of course, the polyols and polysaccharides based on sorbitol, mannitol, and other sugars, lactose, sucrose, etc., small molecules that can be part of the composition. Cellulose derivatives are worth investigating further because they're some of the most common pharmaceutical excipients that are used for tablet binding, controlled release, coatings. So as you can see here, this derivatized cellulose can be in the form of hydroxypropyl cellulose, ethyl cellulose, carboxymethyl cellulose, and so on. So here's a picture of how this hydrated polymers can be added with the matrix tablet. Essentially allows the controlled swelling and even protection of the drug, such that you have a very good passage when ingested, let's say, with the gastric fluids in your body or stomach. Now, starch is easier to talk about simply because we identify them with food. But again, these are very popular polymeric drug excipients. They can be derivatized to specific types of starches, and a number of them have very specific action and counteraction with the drugs that are used to compound them. Talk about chitosan, which is not as popularly used as the former two, the starch and the cellulose. Chitosan actually is a very abundant natural polymer that is obtained by deacetylation of chitin. Now, it is very, very much abundant in nature in that you find it, of course, in shrimp shells, lobster, krill, squid bone, etc. They're non-toxic, biocompatible, and in fact is used a lot in terms of possible applications with implants, as well as different types of more rigid thermomechanical appropriate and biocompatible devices. So of course you derive them by decalcification from the original chitin, and once you decalcify them, you can deprotonate them by removing sodium hydroxide, and then finally, in terms of their compatibility and net charge, the chitosan is the more soluble form, which is neutralized with the acid. So as you can see here, the chitin that is isolated and with the removal of the protein with the sodium hydroxide solution has to be then neutralized by the addition of acid. So it's the chitosan which is considered, which is deacylated, which is considered the more processable form of the polymer. And this is what you'd find in many applications, including their biocompatible surgical applications and implants as well, not only for polymer drug excipients. Now, some of you are probably wondering, when are we going to talk about PEO or polyethylene oxide or even polyethylene glycol? So I will briefly mention PEO. Polyethylene oxide, of course, is a water-soluble polymer that is non-ionic, meaning it doesn't have any specific negative or positive charge that can interact with the DNA or polypeptide, and therefore PEO is in general a very useful polymer that can be used to improve drug efficacy or used as conjugates with proteins to improve their self-assembly. A wide range of molecular weights and groups are available, and of course they are FDA approved. So we'll have more time to talk about PEO and even PEG or pegylation procedures. Suffice it to say, they can be in the form of homopolymers or copolymers, very much commercially available. And in fact, PEO is actually used not only for the drug or pharmaceutical application, but is found in a lot of consumer personal healthcare products that you'd find in many formulations. Silicone. So again, some of you are wondering when are we going to talk about silicone. Silicones have many uses. They can be used for creams, fluids, ointments. They are used for and take advantage of their chemical resistance or stability against various physico-chemical stresses. They are low TG or low glass transition temperature. Therefore, they remain liquid at room temperature. They can be cross-linked to have elastomeric properties. And in fact, they are used as adhesives. And in general, they are non-toxic and they have good biocompatibility as well. Therefore, in many ways, silicone delivers in terms of performance for many applications, including bioimplants. Now, oral drug delivery, again, just briefly touching this part with polymer excipients, means that the easiest way that one can optimize drug delivery is to optimize the ingestion of drugs in tablet or emulsion form. So in many ways, although this has been done with formulations, a more targeted or method for optimizing drug delivery is actually controlled release through encapsulation. So encapsulation means that you have a protective coating layer which allows the diffusion or release of the drug from the core over time. And therefore, encapsulation technologies or making polymers used for encapsulation in microparticle sizes is an important aspect of polymers in medicine. I will not be showing you more about the different types of encapsulation technologies, except that they can be in the form of freeze drying or spraying, or they can be processed as emulsions, dried, and then used for encapsulating various core shell compositions. So talking about PEG, or a polyethylene glycol used for conjugation. PEG, as I mentioned, is quite versatile and neutral, non-ionic. It is very well known in terms of not producing any type of immunosuppression or immune response, unlike the polycations and polyanions. So pegylation or application of conjugated PEGs is very common and useful in different types of drugs, including polynucleotides and polypeptide-based drugs. It can be used to improve circulation time, decrease enzymatic degradation, and provide immunosuppression. Pegylation of different drugs can be used to enhance what we call the EPR effect, or enhanced permeability and retention, also with larger drug molecules or increasing the size of the drug itself. EPR effect simply means that you have more passes or increased permeability because the drug tends to stay longer instead of going faster through the your body. And this has been used very commonly in the design of cancer drugs. And the PEGs can be in the form of linear or star-shaped materials, but certainly chemistry of PEG is very important. So as you can see here, different types of PEG derivatives, which can contain acid, imide groups, thiol, sulfone, azide group, which is very important for click chemistry, alkyne groups. All of these type of functional groups in different PEG oligomers or polymers are used for conjugation chemistries. So as you can see here, conjugation chemistry means that you are able to apply the PEG in various forms, either as graphs or blocks or different types of core shell systems, where again, the PEG typically allows the drug or the particle to circulate longer because of its stealthiness. In other words, the PEG does not cause immune response or is able to go faster through your plasma or even at the right size through the blood-brain barrier. So PEG polymers is very common in medicine, also for different types of tailoring of application, including improving self-assembly. So expect that PEG will be here to stay. However, other types of polymers are beginning to replace pegylation systems. In particular, those that are produced by polycation polymerization that have also seen very good efficacy and a neutral interaction with the body. So I'm going to skip this slide and I'm going to switch to what we call shape memory applications of polymers. So shape memory has to do with what we can observe with the stimuli response that results in a mechanical or thermomechanical change in the shape, the size, upon the application, let's say, of heat. Shape memory polymer simply means that the initial shape was formed during the fabrication or formation, and that with temperature and time, it is restored to the final shape or the original shape. Now, heat is just one way of producing that response. It could be in the form of light or even a chemical environment. But shape memory polymers, needless to say, are very important, especially for implants or biomedical application. Now, I know it's not a polymer, but when we talk about stents or drug-eluting stents that are used for surgery, those are actually shape memory materials, but they are made from nitinol or metals. In other words, polymers that can be used for surgery and tissue engineering will make its way even more compared to their metallic cousins in the future. So just to show you some of these shape memory properties, as you can see here, the shape memory effect involves the control of the cross-linking density on the original polymer, typically an elastic and a viscous component is part of the original composition, which can be changed with the application of a proper stimuli. As I said, this could be in the form of heat. It can be also in the form of UV light, or it can be in the form of a chemical response, meaning it responds to a small molecule or diffusion of gases. So as a shape memory material, it's capable of actuation. In other words, it will stretch or it will compress. And as you can see here, in terms of its possible surgical application as a suture, it's able to close the loop or the catenation of the ring or knot, formation of knot more easily as a shape memory material. Now, we cannot, of course, go forward and mention polymers in medicine without talking about hydrogels. Now, again, because of the broadness of our topic, we cannot give very specific system, although a lot of hydrogels, people can be familiar with natural polymers, polysaccharides, gels, for example, other types of xanthan gum or guar. These are gels. On the other hand, hydrogels can be in the form of PEG, polyethylene glycol containing hydrogels, polyvinyl alcohol. These are synthetic polymers that can be cross-linked. So a hydrogel essentially is a combination of either a physical cross-linking or a chemical cross-linking. A physical cross-linking means it's reversible. A chemical cross-linking is an irreversible component, meaning you form or reform a covalent bond. So just to show you what a hydrogel would do, and again, hydrogels in a sense here can be used for the polymeric drug excipient, drug delivery, as well as scaffolds for tissue engineering. So a physical gel simply means that this is made of phase-separated crystallites and amorphous content. It could be very specific helix or formation of crystalline phase or entanglements that are afforded by stimuli, meaning by heating, cooling, or change in pH, or even the salt concentration. On the other hand, a chemical cross-linking can actually be introduced to improve the formation of a gel. And this can be in the form of esterification, amidation, or click chemistry. To one extreme, we can see here that a epoxy reaction, which is actually a thermoset, starts as a chemically cross-linked gel, which over time and with lots of cross-linking produces a thermoset or a very hard material. On the other hand, a polysaccharide or a polyvinyl alcohol can easily be cross-linked by the introduction of glutaraldehyde, which can be activated and cross-linked chemically to produce a more durable hydrogel. Okay, biodegradability. Is it the same as biocompatibility or bioresorbability? And is it used for drug release or tissue engineering or both? So biodegradable polymers are very important because once they are used, they are expected to go away. Therefore, in bio implants, they can be used as scaffolds for tissue engineering. They can be used as sutures or things to improve wound healing, or they can be used as bioresorbable polymers to induce bone development or the grafting of osteoblasts and fibroblast cells, or the formation of a very compatible and friendly extracellulose matrix or ECM. So talking about bioresorbable polymers are good for devices, prosthesis devices that degrade over time. And in biomedical engineering, there's a lot of interest in this, or even 3D printing, because let's say with 3D printing, one can create scaffolds that will allow the formation of, let's say, tissues or bone of specific shapes or sizes. So things that degrade or typical biodegradable systems can be in the form of polylactic acid, PLA, polyglycolic acid, polydioxanone, polycaprolactone, other types of polyesters, and so on. So they are expected to initially have the shape of the tissue or implant, and then over time they're expected to degrade and go away safely as a biomaterial. Now, let's talk about polymer drug conjugates. Polymer drug conjugates are means to improve the delivery of drugs. Again, pegylation is a common method. Pegylation on, let's say, a polypeptide drug, or the use of polyhema or other types of polyacrylates. These drugs can be made more effective, less toxic, better diffusion properties, and of course the right pharmacokinetic strategy. So as you can see here, these are some strategies for drug conjugation. Typically, these are reactions like the click chemistry or thiolene chemistry, or even esterification, that are meant to be simple, high yielding. On the other hand, some of these conjugated drugs are expected to be released and broken off from the host carrier through enzymatic or hydrolytic action. That is, the polymer already served its purpose simply by improving the pharmacokinetics or in vivo pathway of a polymer drug, or including it in a polymer scaffold. And again, this is a very, very broad tutorial. Bioconjugate chemistry can be done with click chemistry, which is typically involves an thiolene or a cycloaddition reaction with a copper catalyzed alkyne acid chemistry. And this can be made, or conjugation can be made in the form of linear blocks, self-assembling surfactant-like structures, branching, and conjugated pathways. And not enough time, but I can just simply mention polymer protein conjugation as a very important and hot topic. This is not meant to be exhaustive. This is just to show the representation of polymer companies, drug companies that are interested in the development, use of polymers in medicine and pharmacokinetics. And some of them might be familiar as some of your sources for your starting materials, and also quite used in manufacturing. Now, unfortunately, we don't have much time. I was going to insert some of our work on biosensing, where we actually try to model enzymatic receptors, protein enzymes and receptors, simply by producing a molecule or supramolecular structure or film that mimics the interaction between a substrate and an enzyme. And we do this simply by doing what we call a lock and key model, where we actually synthesize the lock based on a key or a targeted biomolecule or chemical, and then simulating the cavity by synthesizing that cavity through cross-linking reaction. So we have actually developed a molecular imprinting in our lab, primarily by using electrochemical methods. So the monomers we've developed typically surround the molecule, electropolymerize them on a substrate, and do transduction studies. And with the remaining time, I'm just going to show you an overview of how this works. Typically, the molecule of target, so these are specific drugs that we've targeted, from caffeine to theophylline to naproxen. We can surround them with monomers based on the thiophene or terthiophene series, electropolymerize them. In fact, the complexation of the drugs with the polymer can be modeled in order to improve the efficiency of the cross-linking process. And we do this by ab initio calculation. So with this method, we were actually able to use or develop a film or a coating that specifically senses drugs that are introduced based on the template or original template, having the higher sensitivity and selectivity because they were the original template compared to the analogs or other molecules that were not templated. So here we have a very high response with surface plasmon spectroscopy based on the presence of the originally templated drugs. We have actually utilized a technique based on molecular imprinting as well as definition of arrays to differentiate chiral sensing between norepinephrine. Norepinephrine, of course, only one form is active, one enantiomer, as an amphetamine. On the other hand, the other is not. So can we use electromolecular imprinting to do this? So we have developed this sensor, and what we have shown is, again, depending on the analog and the original template, we are able to distinguish the two enantiomers from each other, including differentiation between naproxen and dopamine. And in fact, this technique has been widely used in many other chemicals, and we've published this since, to detect sarin, dopamine, bisphenol A, melamine, morphine, aspartame selectively. More recently, we have actually used this type of strategy to detect proteins coming from the dengue virus by utilizing molecular imprinted templates of epitopes or oligopeptides that are typical of what would be isolated from a dengue virus. And we have used quartz crystal microbalance to quantify the sensitivity and selectivity of these drugs. Lastly, I'm just going to let you know of a collaboration I've had with the Steinmetz group, who had since moved to UC San Diego, where we actually work with them to conjugate and to improve the efficacy of viral delivery systems. In this case, these are plant viruses like the cowpea mosaic virus or the tobacco mosaic virus that can be conjugated with drugs or can be used as delivery systems. And indeed, these are actually polymeric assemblies or systems based on polypeptides that can be genetically engineered for medicine. So with that, I'd like to thank you and appreciate your patience, and I'll be happy to answer any questions. Thank you.
Okay, thank you, Dr. Advincula. We do have one question so far, and for any of You others out there that have a question, feel free to type it in to the questions module or raise your hand with the raise your hand button, and I will call on you shortly. The question we have is, if it is possible, could you talk about common manufacturing methods for polymers in the biomedical field?
Yeah, let's say with drugs, with devices, let's say, and even with 3D printing, this could be very different processes. So with drugs, it's basically a formulation strategy. So they work on emulsion, freeze-drying, tablet preparation. So all those things means that you need to have a formulation optimized and made into the right liquid or tablet form. When it comes to devices, we can be talking about injection molding, different types of formative manufacturing to create the exact shape. And some of these methods based on extrusion, thermoforming, etc., are well known in the plastics field. For me, actually, the high interest is on 3D printing, because with 3D printing, various types of polymers can be adapted to techniques like FDM, SLA, SLS, and prepared in the right shape. In fact, there's a lot of interest on combining imaging, let's say with MRI or PET scanning, taking that digital file and directly 3D printing the material based on the desired implant material or resorbable material.
Great. Is there anyone else with a question? Please raise your hand or get it in shortly. I don't see any. Oh, wait, I think we've got one coming in here. Okay, so let's see. The next question is, can you please explain the term plasmonics, what it actually means and its application?
Yeah, thank you. I like that question because plasmonics certainly is used a lot in nanotechnology terms. Plasmonics typically are associated with gold, silver, or other types of metals that produce plasmons. This can be measured as localized plasmons or guided wave plasmons. So what plasmons are basically is easiest term is called surface light, quote unquote. So these are ions, electrons that are on the surface of this, let's say, noble metals that can be observed visually as a change in color, or in terms of what we call ATR measurements as a change in the dielectric properties, resulting in a change in reflectivity. So gold plasmons or use of silver or gold nanoparticles specifically are used for theranostic applications, meaning they can be bioconjugated and used for labeling, where these nanomaterials can be targeted to specific tissues and then easily identified by, let's say, microscopy. Or some type of gold nanomaterials can actually have a property by absorption of near-infrared light, which passes through the skin and therefore can be used for heating. So we've had the phenomena of plasmons actually used for surgery or improving surgery or healing, simply because they can be activated by heat.
Okay, and we've got one more question here. What are the main disadvantages of the current software for designing and simulating mechanical properties of polymers? For example, researching porosity for scaffolds or shape memory material.
Yeah, the question is a little complicated. I'll try my best to answer it. In terms of simulation, of course, well, one software that I can suggest is COMSOL, which is applicable for many things, and I cannot suggest other types of specific software for simulating thermomechanical properties or measuring surface area. Unfortunately, I may not be the expert on that, but I'd be happy to go over the questions by email. But certainly the properties you mentioned, whether it's the thermomechanical properties or porosity or surface area, are very important measurements. Empirically or testing-wise, one can prepare these different blend compositions or materials, let's say polyether, ether ketone, which is a high-performance polymer versus that of a polycarbonate or even an elastomer. We can use tests to design to measure the tensile properties, the modulus, the flexural strength, the hardness, and those are very common in the plastics world. On the other hand, porosity can be measured in terms of BET surface measurements, in terms of what we measure with the surface tension, interferential tension. We typically use other types of measurements based on contact angle and so on. So empirically, experimentally, there are many ways to measure these properties. On the other hand, simulation or software, unfortunately, I'm not the best expert on that, but I'll be happy to communicate with you on those matters.
All right, I will take this opportunity then to ask one final question, which is, can you give us a brief preview of next month's webinar topic, smart proppants and oil field productivity?
Yeah, this is quite different from our topic of today. And interestingly, of course, the common trend is we're still talking about polymers, in this case, used by the oil and gas industries. They're called resins or propant resins that are used to improve the property of this tool, quote unquote, or material introduced in oil wells or well completion. So I'll be happy to see you then if this is your interest. If not, I'll be happy to communicate with the rest of our audience and attendees by email regarding the topic today on polymers in medicine and pharmacy.
Okay, thank you, Doctor. Please join us for that session if you're interested on Wednesday, July 17th, once again at noon Eastern Daylight Time. And thank you all for joining us for this session of the Park Systems 2019 Materials Science Research and AFM webinar series. You can find more information about Park Systems AFM at parksystems.com. And please direct any AFM questions you have to inquiry@parksystems.com. If you have any questions specific to this webinar series, feel free to reach out to me directly at richard@parksystems.com. Thanks again, and we'll see you next month.